Okay, welcome everyone. My name is Michael Downey, Library and Information Services Coordinator at the AWRI and moderator for today's webinar. It's my pleasure to welcome you to today's session on optimising the management of soils and organic matter and the role of soil in carbon drought resistance. Now, to enlighten us on this topic, we're lucky to have with us today Dr. Jeff Baldock from CSIRO. Jeff leads the Soil, Carbon and Nutrient Cycling Group in CSIRO Agriculture and Food and has been studying soil organic matter chemistry, dynamics and its contribution to soil productivity, including the provisions of nutrients to growing crops and pastures for more than 30 years. Jeff led an Australian research program aimed at defining the influence of agricultural management practices on the quantity and composition of soil organic carbon, and continues to lead and contribute to a range of research projects that are improving Australia's national greenhouse gas inventory, as well as developing methodologies for inclusion in Australia's Emissions Reductions Fund. More recently, his research has extended to the quantification of organic carbon stocks in blue carbon environments, including mangrove, salt marsh and seagrass environments. Now, before I hand over to Jeff, to tell us more and get us underway, a couple of quick notes on what to expect for those that are new to webinars. The format for today is a short presentation followed by a Q&A. The webinar is being recorded and will be available to view via the AWRI website. It's also important to remember that webinars are interactive and that there are a number of ways you can get involved. To ask a question, please type into the question section of your control panel in the bottom right corner of your screen. For those with access to a microphone, there's also the option of speaking directly with our presenter today by clicking the raise hand button. This is located in the top left section of your control panel. Please send through your questions at any time, and once we get to the Q&A, I'll get as many questions through to Jeff as possible. And just finally, for anyone that would like to get involved through Twitter, please use the Twitter handle @_AWRI. Okay, that's enough for me for now. I'm now going to hand over to Jeff to get us started.
Okay, I think it should be up now, Michael. Yep, good. Thanks very much for the opportunity to come along and give this presentation today. The work that I'm going to talk about I need to acknowledge that it's really come about from some funding that we've got from the Australian government and from the Grains Research and Development Corporation. And what I'm going to be looking at today is thinking about what organic matter in soils is, what the potential role for it is in drought resilience, and I've also added a couple slides towards the end about the role of organic matter in plant nutrition as well. So the outline for the talk is we're going to basically follow the following. I'm going to look at, first of all, why are we even interested in soil carbon? I want to talk a little bit about what it is, both organic carbon and organic matter, and how those can be used, and what you need to think about if you're getting analyses done. A bit about the role of organic carbon or organic matter in soil, water, and water use, and in plant nutrition. A couple of comments at the end about where and how we might be able to increase soil carbon, then I'll just finish up with a quick summary. So let's get started. So the first bit is that I wanted to go through is why we're even interested in organic carbon at all, or organic matter. And I might say, just to start this, when you send a sample of soil off for a test, you'll get a result back and that result should tell you whether it's organic matter or organic carbon that they've actually given you the numbers back for. Just remember that organic carbon contents are about 1.7 times smaller than organic matter, and it's important then that you distinguish which one the analysis lab is actually giving you, because there is a factor of 1.7 between the two. So organic matter equals 1.7 times organic carbon. The first reason we should be interested in carbon in soils is because of the potential role that it and the organic matter that it's contained in can do for soil productivity. Organic matter has beneficial functions on soil biology, soil physical properties, and chemical properties. And just as a couple examples, in the biology space, It serves as a source of energy. It holds a lot of nutrients, and it provides the soil with some resilience against change, or an ability to respond positively if put under a stress. From a physical point of view, organic matter is important in infiltration, stability of structure, and water holding capacity. And on the chemical side, it contributes cation exchange. It can buffer pH change. So it's a component of soil that contributes to a wide range of properties. Today, our focus is going to be on the sort of water side and a little bit on the nutrient side. The other reason we're interested in soil carbon these days is because of the potential of soils to sequester carbon dioxide and reduce net greenhouse gas emissions. And if we take this plant soil type system and think about what the carbon cycle in it is, and the plants are picking up CO2 from the atmosphere, converting it into leaves, stems, roots. As that material dies, it falls onto or into the soil and becomes soil organic matter through decomposition. We can also bring materials from off-site and add them to the soil to create soil organic carbon or organic matter. At the same time as all these inputs may be happening, the other thing that can occur is we'll get in a series of losses. Not all of the carbon taken up by the plant is actually retained. Some of it goes back and is respired back to CO2 without even getting to the soil. All the time, microbes in the soil, just about all the time, microbes in the soil are active. They're mineralizing, respiring carbon back to CO2. We also take a product away that has carbon, and we might harvest up residues and take them away. So when we're looking at the net effect of our plant agricultural systems on soil carbon values, we really got to think through the balance of inputs and losses. If we want to build carbon, we've got to shift that balance so that we're putting more in than is being lost, and gradually then we'll build carbon through time. If we can do that, then there is opportunity to lift our soil carbon contents. By lifting and maintaining those soil carbon contents, then we would basically reduce the amount of CO2 in the atmosphere. And if you were inclined to, there are some opportunities you might want to pursue around greenhouse gas emission offsets programs. And the Emission Reduction Fund of the Australian government actually has a soil carbon component in it now. Okay, the next thing I wanted to go through is, and have a bit of a think about, is what soil organic carbon actually is. What's it composed of? And if we think about a system, we're looking over a paddock or a vineyard, and we're thinking and looking at the different types of carbon that are in this system. And I'm not talking about the live bits here, I'm just talking about the residues and the stuff that's in the soil. We'd see plant residues on the soil surface. We'd also potentially see plant residues buried in the soil, bits and pieces of leaves and roots that we can actually see, typically stuff that's bigger than 2 millimeters. In addition to that, though, as the materials are decomposing, the size decreases, and we move into something that we call particulate organic carbon. And by definition, that's carbon that has a size, or is associated with soil particles that have a size of 2 millimeters down to about 0.05 millimeters or 50 microns. When we move in, get smaller still, we now talk about soil humus, the organic carbon that's associated with soil humus, and our definition for that is that it's carbon that has a size less than 50 microns or 0.05 millimeters. When you move through these different types of residues, what we find is that the materials up towards the top, the plant residues, tend to be high in carbohydrates. That CH2O is a short form symbol for carbohydrates, things like cellulose. So they tend to be the energy rich. They have a lot of energy available in them for microbes, and the microbes use that during decomposition. As you move down through that sequence, the decomposability or the recalcitrance increases. Decomposability decreases, recalcitrance increases. Down towards the bottom end again of that sequence, we tend to see that they're nutrient rich. So these different types of materials are actually serving different purposes in the soil and on the soil. That's one reason why we think it's really important not to just be thinking about soil organic carbon as a single number, but start to think about what it's made of. One other type of carbon that we found in soils, in particular in Australian soils, is something we call resistant organic carbon, and that tends, in the Australian context anyways, to be dominated by charcoal-like materials. It's not the charcoal that's been produced by European settlement in Australia and by our current agricultural practice, although there may be some from that. The majority of the charcoal that we see is basically been produced potentially thousands of years ago and is accumulated in the soil because it's relatively resistant to decomposition. Now, when you send a sample off to a lab and you get a soil carbon analysis done, you're basically going to be looking at the three fractions that are highlighted in the blue box. The stuff at the top we generally don't include, and part of the reason for that is because it's very decomposable and labile. By including it, in particular in the carbon accounting systems where we include it, we just bring a bit of volatility into the carbon accounting process. So we tend not to account for that when we're looking at soil carbon. If we were doing nutrient budgets and things like that, we would account for it, but we'd keep it in a box separate from the soil carbon box. Now, just to give you a bit of an idea what these things look like, these are some electron microscope pictures of some of this material. The particulate carbon, you can still see residual bits and pieces of plant structures and tissues. It still looks, although it doesn't look exactly like a plant material, you can tell that it was derived from plant material. When we move into the humus carbon, which is the stuff less than 50 microns, that structure's gone. We don't see that. Most of what you see here in terms of the angular shapes and stuff are the soil particles. The organic matter serves, in this case, more as coatings. These little bits and pieces hanging off the sides might be organic matter, but typically it tends to be associated with the mineral surfaces. That last fraction that I talked about, the resistant one, or the charcoal-like stuff, this picture is every particle in here is charcoal, and one of the things to notice is how small these particles can be. They can be less than a micron. So when we're looking at soils that have a lot of charcoal in it, often you can't see the charcoal. They just end up making the soil look a little bit darker. Some of the highest carbon charcoal containing soils in Australia are from our old grasslands, where repeated burning of the grasses has resulted in the production of these fine little bits and pieces of charcoal that we wouldn't see just looking at the soil itself. So I wanted to just briefly talk about how we isolate these different things. The first step is a fairly easy one. We'll sieve the soil to less than 2 millimeters, and then we disperse it and we pass it through an automated sieve shaker. We get a coarse and a fine fraction, and again, just to show you what these things look like, in those coarse fractions you'll still see little bits and pieces of plant debris. In the less than 50 fractions, though, that's gone. We can't see that anymore. It's too small for us to see without the aid of microscopes. When we want to work out how much charcoal or resistant carbon is in this, we take these samples off and we put them into a spectrometer called a nuclear magnetic resonance spectrometer, and that can allow us to identify how much of the carbon associated with those materials actually fits into this resistant fraction. Now, why are we interested in this soil carbon composition? Why do we think it's so important? This is some data from 6 sites from New South Wales. We went out to the sites, collected the soils, came back and measured the carbon contents. And at this stage, all I've done is presented the carbon content on this axis against the name of the location in New South Wales where these were taken. And at this stage, we'd know that this soil is high. Oh, I should have mentioned, the axis here is milligrams of carbon per gram of soil. If you think more of carbon in terms of percent, you just divide these numbers by 10 and you'll get the percent carbon of the soil. Right now, with this data, all we can really say is that this soil out at the beginning has a very high value, the soil down here has a very low value. If we were worried about how vulnerable that carbon was to change, how stable it was against decomposition, or what its contribution to different soil processes is, it's going to be very hard to make any inferences about that. So what we've been doing is looking at what is that carbon composed of. So what I've done here is basically it's the same graph. I've just broken up that red bar into the allocation of the carbon to those different fractions. So the green is the particulate, the blue is the humus, and the black is the charcoal or the resistant component. What I now can see from this soil here, this first one, although it had the highest carbon content, it also had the highest fraction of its carbon allocated to that particulate component, and that's the most degradable. So if I was looking at assessing, should I enter that soil into a carbon trading scheme, I'd have to be tempering my decision with the fact that almost half of the carbon in that system is highly degradable, and I might not want to put all of it into the system. Conversely, I come down to the soil that had the smallest amount of carbon, and I say, oh, well, there's not that much of that PLC. It's only a small component of the total carbon in that system. It's quite different then in terms of its lability going forward. And one of the things we're looking at, and we haven't, well, we're hoping to move forward with suggesting that we can get an idea of the vulnerability of soil carbon to change from these fractions, for example, by expressing the particulate organic carbon as a function of the humus, the sum of the humus plus resistant. I'll speak a little bit more about why I think these fractions are important to soil functions in a minute. So the next thing we did was look across a broad range of Australia's agricultural soils, and we've measured how much carbon is in these different fractions. And on this figure, I've presented a relative frequency. So out of the 312 soils, this is the distribution of the soils in terms of their particulate organic carbon content. So it's telling us that on average there's about 20% of the carbon in these 312 soils was related or ended up in the particulate fraction. But notice that there is a very big range. If I do the same thing and put humus carbon up and then the resistant, the point of this slide, I guess, in the take-home message is that not all soils are the same. That the allocation of carbon to these different fractions can, and the variance in that can be pretty significant when you move from soil to soil across the country. And we think understanding that is going to help us understand a lot more about how carbon is influencing soil properties and functions. Now, running through that fractionation process is a pretty complicated and expensive process. It's probably really only going to be, at this stage, the laboratory work of doing it. It'll be a research practice. But one of the other things we've been working on is how can we come up with a method of making our understanding of fractions and quantifying those fractions available to growers to understand what's happening in their own soils. So we've been working on trying to develop a more cost-effective way of looking at this or completing these analysis, and we've been using mid-infrared. So MIR stands for mid-infrared. What that does is it gives us a spectrum, a mid-infrared spectrum of the soil, and it tells us in essence what the chemical bonds that exist in that sample of soil are. We then use a partial least squares regression approach. It's a statistical approach that lets us go and look at those spectra and get information out of them about a particular variable that we're interested in. In this case, we want to know, can we use the spectra that we get from an MIR spectrometer to predict how much of the carbon in a sample, well, how much carbon is there in the sample to start with, and how much of it can be allocated to those different components that I've been talking about, particulate, humus, and resistant. So for this technique, it's the beauty of it is it's relatively simple and easy. We place some soil in these little cups, just a close-up of the cups with the soil in them. Those cups then go onto the spectrometer. We run it through the spectrometer and we get the spectrum, the MIR spectrum. So this process, I'll back up. The lab process that we run through takes weeks and lots of expensive equipment. The MIR spectrometer is not cheap. It's still relatively expensive, but the process of getting the samples is probably about 6 minutes of grinding plus 3 minutes maybe of analysis. So you're looking at a much more reduced analytical time to process and get some data out of it. Using those same samples that we've collected from across the country, we then tried to derive relationships to ask, can we predict the amount of carbon in these fractions in these soils from infrared? And this is the results that come out of this partial least squares analysis. So I've got, in this case, this is the total organic carbon content of the soil. It's square rooted to try and help out with the linearity of the equations, but in essence we've got a measured value here and we've got a predicted value here. What we would like to see, and what's happened, is we get a nice linear relationship. There is some scatter. Not all soils are exactly the same, but we've got a reasonable relationship. We've been able to derive an algorithm that can take an MIR spectrum and give us a reasonable prediction of what the organic carbon content in that soil is. We applied the same approach across the particulate, the humus, and the resistant fractions, and we've actually ended up then with a method that gives us the capability to not only predict the organic carbon but the allocation to those fractions. One of the things we're looking at now, and the challenges now, is how do we make this whole process rigorous and potentially deployable out into the agricultural analytical labs. So now I'm going to move into thinking about drought resistance and plant nutrition and the role of organic matter in that system, in those two properties of the soil. I want to start though by just thinking about water in the soil and the way it moves and where it comes from. So if we start with this little picture, we've got, it's raining or we've got irrigation, it falls onto the soil surface. That water will either infiltrate. If the infiltration rate on a particular soil is not high enough compared to the rate at which the water is falling, we'll get runoff. As the water enters into the soil, it fills up the soil water holding capacity. If there's too much water and it completely fills that, we'll actually get drainage out the bottom. Now once we start looking at where that water goes, if there's no plants or there's bare surface, that water can be lost through evaporation directly from the soil, or it can be lost through transpiration if we've got plants there. In our agricultural systems, what we want to try and do is maximize the loss of transpiration and minimize the loss of evaporation, minimize runoff, and minimize drainage. And if we can do that, we're making our systems more water efficient. So if we want to drought-proof our soils, the things we've got to be thinking about is how can we minimize the losses through evaporation, runoff, and deep drainage, and maximize the fraction of the water that we get that can move through the plant through transpiration. So that's basically what I've just said. So creating drought resistance, we minimize evaporation, maximize infiltration, and maximize the capacity to store. And if we do all that, we'll minimize the losses. Organic matter and organic carbon in soils can contribute to all of these, all three of these requirements to enhance drought resistance. Firstly, as a surface cover with plant residues, we can in some soils we might be able to improve infiltration by reducing things like surface slaking and sealing caused by raindrop impacts. We may also be able to reduce evaporation if we've got a good mulch layer over the top of the soil. We can stabilize aggregates and pores with organic matter and improve the ability of the soil to hold water. If we can do that, then the other things we'll do is reduce the amount of runoff and drainage. So when we're thinking about drought proofing, organic matter has some potential roles in all of the aspects that will make a soil more drought resistant. In the next couple of slides, the picture of the water balance, if you like, is going to stay constant. And I'm going to be just stepping through a few examples of how organic matter or organic carbon can contribute. Now these examples are individual examples for particular soils in some cases. They are just examples. The numbers that you'll see on these slides may or may not apply to a particular soil, to your soil. We would need to have to think about that. The first property that I wanted to look at was bulk density, and if we're looking at soil bulk density, we're also looking at total porosity. But there's an equation out there that says bulk density is equal to 1.608 times minus 0.078 times the percent of organic carbon. And if you plotted that, you'll get a graph something like this, where we've got bulk density on the y-axis, we've got carbon content along the x, and that equation in this case is a straight line. Other equations that people have come up with aren't necessarily straight lines, but what this is basically saying is that for every % of organic carbon in the 0 to 10 centimeter soil layer, we'll get an extra 3.3 millimeters of pore space, and therefore potentially 3.3 millimeters of water holding capacity. And whether that's plant available or not, I'll talk about in a minute. So with these bulk, because we've got the bulk density, and there's a relationship between bulk density and total porosity, that's what lets me calculate this total pore space and allows me to make this calculation of how much extra water that soil might hold in response to having a higher soil organic carbon content. The next factor I wanted to look at was infiltration rate. In this case, the data that was presented in the paper came from forests and natural succession and arable land. It's an extreme example, but it makes the point that as we enhance soil carbon stocks, our ability to infiltrate water into the system may be going up. In this case, definitely went up. We've gone across some very different management or very different systems here, but basically as the carbon stock went up, the rate of infiltration went up. So that would say, if we're getting a rainfall event, more of that water has the potential to get into the soil and less of it will run off when we've got the high carbon stocks relative to low carbon stocks. The last point about water that I want to make and the role of the potential role of carbon has to do with how much of the water that a soil can hold is actually plant available. And when we look at the amount of water that exists in the system and we go across different textures, so increasing clay content, there's two parameters that are of importance. One is a thing we call the lower limit, which is how much water a soil still holds after the plants have been able to get everything they can out of it. And as clay content goes up, the amount of water that remains in the soil after plants have pulled everything they can out also goes up. The upper limit is the amount of water that a soil can hold against drainage. So the difference between these two is what we call plant available soil water. Now, just again as an example of the role of organic matter in this, some predictive functions have been put together. I'll explain what these mean, but this is basically saying that the volumetric water content of the soil can be described by an equation. The parameters A and B in that equation can then be described or can be estimated from a series of properties of soil: the clay content, the organic carbon content, and the bulk density. Because organic carbon features in these two equations, then organic carbon is actually changing the way in which these relationships, lower limits, can respond. We can use this equation then to get the practical application of that is, if we, in this case, we've gone and taken data from about 40 red-brown earths or chromosols from South Australia. We've used those equations to predict the upper and lower limits and to define what the change in water holding capacity of the soil was, plant available water holding capacity, in response to boosting the organic carbon content by 1%. So let's say going from 0.5% organic carbon to 1.5% organic carbon. And what this line then shows is that as the clay content is going up, the change in carbon and the change in water holding capacity induced by a change in carbon is decreasing. And that's because in the higher clay content soils, clay is having a big role in defining the actual amount of water a soil can hold. Organic carbon is still important, but not as important in the sandy end. So at the sandy end of this, we're saying for every % increase in organic carbon that you might be able to achieve, we'd be looking at, say, 4 to 5 millimeters of water holding capacity. At the more clay end, or 30% clay, that value drops to 2. So if we said on average we had an extra 3 millimeters, then what we'd be saying then is, and this sort of is, I put this slide together for some grain production work in the past. If we had 3 millimeters of extra storage and we had 10 rainfall events over the year and we held that 3 millimeters, then we'd say in total we'd have an extra 30 millimeters. That translates roughly using some generic relationships for grain production into about 600 kilograms of grain. Now, because we know that it might result in that, we can start putting a value on how much that increase in soil carbon is actually provided. The one issue to think about, though, and to bear in mind, is it's much harder to increase the carbon content on a sandy soil than it is on a clay soil. So if we were out in the Mallee on a sandy soil with 0.5% carbon, to move that to 1.5% carbon would be a big challenge in terms of trying to get the carbon values to move that much. Okay, the next thing I wanted to move on to and is related to the nitrogen cycle and the provision of nutrients to growing crops from organic matter. So just to recap a little bit, we've talked about the composition of organic matter, we've talked about the role it might have in soil water, and now I want to look at the role it might have in providing nutrients to growing plants. So if we've got our plants, they have a bunch of nitrogen in them. That when those plants die or they drop residues or roots slough off, that organic material associated with the residues turns into soil organic matter. And the organic matter, although we typically measure it as organic carbon, contains organic nitrogen. And 95% of the nitrogen that you'd see in the soil, at least 95%, often much more than that, of the nitrogen that exists in a soil is actually in this organic form. And that's not really accessible to plants. What also happens is as the microbes are chewing up these residues, they are actually converting and through when they die, their residues also will go into creating this soil organic nitrogen pool. To get that into the plants, the first thing that has to happen is that organic nitrogen has to decompose and produce inorganic N, nitrate and ammonium, or maybe some soluble organic N. And that's I'll come back to the immobilized in a second, that's the form that the crop or the vines or the plants that are in the system can take up. The interesting thing is that because this is an active system and the microbes are there all the time and the plant roots are there, everybody's competing for N. And it is possible for the microbes, if they're chewing on a really labile carbon source and don't have enough nitrogen, they will scavenge nitrogen from the system and they'll compete with the plants. So we've got this process called mineralisation, which releases nitrogen to this plant available pool. But there's also a process called immobilization where that nitrogen is soaked up by the microbes temporarily. It'll eventually come out again, but the microbes are reducing the size of the plant available pool. And I'll go through how that happens in a minute. What we try to do to make sure the plants get enough N is augment that supply that can come from the soil naturally with fertilizer. We also have the potential in some of our agricultural systems to get symbiotic and non-symbiotic fixation, contributing nitrogen into the soil. And we also have mechanisms by which that nitrogen can be lost, leached, denitrified, or volatilized. And finally, we can have that nitrogen removed in products. So what I wanted to look at a little bit now, and when we think about the potential role of organic matter in provision of nutrients, it's really about how much of the nitrogen in this case is being mineralized and released into this pool, and how much of it's being immobilized, how much we can create over the course of the year, and how much, say, fertilizer do we need to add or to augment the supply in order to make sure that our plants are growing as well as they can. So one of the big questions when we look at defining fertilizer application rates is how much nitrogen are we going to get out of the soil during that growing season. And one of the big controlling factors on soil N supply from organic matter is the carbon to nitrogen ratio of the material. And I'm going to run through just a couple little calculations here. The numbers that I'm using, I've kept like 100, kept them pretty easy so that the calculations are obvious. In reality, something like wheat residues might have a C/N ratio anywhere from 50 to 85, maybe even 110 in extreme circumstances. But just note that these numbers are not, I'm not saying that all wheat residues have a C/N ratio of 100. I'm using 100 because it makes the calculations easy. When that wheat residue decomposes, and what are we doing? Well, we're decomposing that residue and we're turning it into soil organic matter. When that happens, on average, about 70% of the carbon will blow off as carbon dioxide. which means from that original 100 kilos of carbon, I'm moving about 30 kilograms of carbon across and making it into soil organic matter. Now, back here in this wheat residue, having a C/N ratio of 100 means for every 100 kilograms of carbon of 1 kilogram of nitrogen in that residue. If I move 30 kilograms across and I'm making soil organic matter with a C/N ratio of 10, then I need 3 kilograms of nitrogen for that process to happen. The issue is that I've only got 1 in those residues, so what's going to happen, the microbes in doing this decomposition are going to scavenge nitrogen out of the soil. So they're going to compete with plants and soak up some of that available nitrogen to allow this process to occur. If on the other hand, this is the numbers associated with that wheat residue, now I'm going to look at what happens if I've got, say, a pasture legume residue in place, which has a C/N ratio of 100. Following all the same calculations, because it's got a C/N ratio of 100, I've now got 5 kilograms of nitrogen. When I convert that 30 kilograms of original carbon into soil organic matter, I still only need the 3, but now I've got 5. What happens is I release 2. So you'll often see in broadacre agriculture or here, when people are following big cereal crops, they need to put a little bit of extra N out. When they're following big pasture crops, they might pull back on their nitrogen supply. And it's basically because of this balance between immobilization, which occurs during the wheat residues, and net mineralization, which occurs when we've got things like metic residues. Now the next bit, we know that soil humus has a C/N ratio of about 10. So that means we've got 10 kilos here, and if I run through all the same calculations, I'm actually going to release 7 kilograms. So the soil organic matter, in particular the humus fraction, does have a high capability to provide nitrogen to growing crops as it decomposes. And in fact, as soil organic matter values have fallen since we've cleared land, one of the things that we've done is actually mineralize and mined a lot of the nitrogen reserves that exist in soils. If we want to build organic carbon back up again in soils, one of the things to think about is, because this C/N ratio is relatively have to find a supply of nitrogen to allow that to occur as well. Now, how does all this relate to, say, the composition of soil organic matter? And this is a slide that I've put together just to give you an idea of it. And we're starting here. We've got our PLC, our HOC, and ROC, so the particulate, humus, and resistant fractions. I've given them a C/N ratio. These soils are fictitious, so I've put them together to demonstrate the point. Soil 1 and soil 2 have the same amount of nitrogen, organic nitrogen in them, in terms of kilograms per hectare, but it's been allocated across these fractions in a very different way. If I say that these fractions in both soils, the fractions decompose a very similar way, the net result, and when I look at how much nitrogen, the net mineralization, how much nitrogen would be released to a growing plant, for that soil 1 we find we would get out 102 kilograms of N. The soil 2 we would only get 16. We started with the same amount of nitrogen, but because now we know something about the allocation of that nitrogen to these different fractions and how fast those different fractions decompose, I can get a better estimate of what might be coming out. Now again, these are fictitious soils set up just to demonstrate a point, but we're hopefully going to be embarking on a project to actually apply some of this to this thinking and come up with real values from real soils to help us better understand nitrogen cycling and the role of organic matter in providing that nitrogen to growing crops, hopefully then leading to better fertilizer rate recommendations. Okay, the next and last comments that I'd like to make is just about soil carbon content and can it be increased and where and how, and just a few comments about that. The first thing in terms is how can we go and identify where soils or regions exist that have a potential to increase soil carbon stocks or contents. And the first thing to think about then is carbon is a balance between inputs and losses. Probably the best thing to do is to try and identify soils or areas where we know we're not capturing as much carbon as we could in our crop given the amount of water potentially that's available. So where our resource use isn't maximized, what it's basically saying is where are we foregoing yield? Where are we not achieving the upper limits of yield? And if we could achieve that upper limit and grow a bigger crop or more biomass and return more of that biomass to the soil, then that's one place where we could get sort of the double increase of both enhancing productivity and potentially enhancing carbon because we're putting more carbon back into the soil. The challenge there is we can identify those areas, but then we have to decide whether or not that reduction in resource use efficiency can be enhanced by management. In other words, is it under the control of the landowner, or is it something that they have to live with because they're not going to be able to modify? And just a couple of scenarios. If we found a place that we could do this and we wanted to keep the same management system that we currently have in place, then we could do things like maximize resource use efficiency. It might be something as simple as making sure we put a little bit more fertilizer out to allow crops to achieve their upper limit of production. We'd also want to maximize the carbon retention and return to the soil. In a broadacre agriculture, an easy thing to do there would be to reduce any burning of residues that might go on or baling up and harvesting of residues. Try to return as much carbon as you can. And examples of doing that might be liming to reduce acidification, to increase the pH and get better growth, to make sure your fertilizers are right, to consider maybe rotational grazing. Another option would be to shift to an entirely different system. The introduction of perennial vegetation where it's appropriate. Try to keep a green leaf there for as much of the year as you can, because if there's a green leaf there, that means it's photosynthesizing, it's capturing carbon, and it's putting carbon into the soil. We could look at using cover crops. They will have potentially an opportunity cost, but maybe that opportunity cost can be outweighed by the nutrition and the carbon that's left behind. And we can look at using organic manures or green manures, organic amendments or green manures, particularly if we're close enough to a source, an easily accessible source. So in that case, what we're really doing is augmenting the ability of our agricultural system to supply carbon by putting through an external source of carbon. Just a couple of things I think that are really important to say about soil carbon. This is some data that comes from a very long-term experiment in a place called Rothamsted in the United Kingdom. We've got soil organic carbon content on this axis, and we've got years on this axis. They established these experiments back in the 1800s, and they've got soils, and they've kept this experiment running constantly, and still is running. What's happened in this case is we started from a given point. On one treatment, they've done nothing, so they've just let it run through. On another treatment, they added manure, but then they stopped adding manure, and this is what happened to the carbon values. In the third treatment, they added manure, and they've kept adding manure. So the points to make here is that soil carbon changes over long time periods. It's not something that's going to change in a couple of years. We'll be moving. You might be able to detect some positive changes, but it's unlikely that you'll be able to say how far or where is this soil carbon going to go to in the long term just through a few years of analysis. The second point is that most soils, if you make a management change, they have a finite capacity to store carbon. Big changes will happen early, and it'll move towards a new, what we call equilibrium value, which is indicative of the new management system. And the third point, probably the most important, is if you do something to build carbon, you need to keep doing it. Because if you stop doing it, the soil carbon values will start to fall again and come back to where they were. So if you've implemented a practice to build your soil carbon values, you get them up there, and you want to keep them there, you're going to have to think carefully. You're not going to necessarily just be able to stop what you're doing unless you're willing to accept the carbon loss, and maybe you are, and then you could go into another rebuilding phase later. So just to summarize, I guess the main points that I hope I've come across is organic carbon, organic matter is composed of a variety of materials. We've got some techniques that we think break that up into three distinct classes. They are by no means tell us all the variability of soil carbon. There's a lot more variability than those three classes of carbon will hold, but they're hopefully a way that we can get some of this out into the agricultural systems. We've got some measurement techniques that can quantify that composition and hopefully move it towards something that can move into analytical labs across the country. Knowing the composition as well as the content we think is informative. It should help with the vulnerability of soil carbon to change. I think it also is going to have a big role, and sorry I forgot to put that point down, but will also have a big role in defining how much water a soil can hold and how much nitrogen we might get from it. We've got the research community has developed predictive equations for describing the impact of organic matter and carbon on soil water availability. The one, just to be again emphasize, some of them are very site and soil specific, so if you manage to see relationships like the ones I've presented, you need to be careful or to understand whether they will or won't apply to the particular soils that you're working with. We're expecting that improvements can be made on these sorts of things now that we're able to measure these fractions and can relate exactly what's going on with the fractions to what we're seeing in the properties of the soils. And with that, I'll finish up.
Okay, fantastic. Thanks for that, Jeff. We'll move here straight into a Q&A. So if you've got any questions for Jeff, he'll stick around for a while. Please start sending them through. Just a quick reminder for those that are not familiar with how to send in a question, you can type into the question section of your control panel, which is in the bottom section of that panel, or you can click the raise hand button if you've got a microphone and you want to chat directly with Jeff. Just got one comment, or yes, something from Darren Fahy just saying hi to you, Jeff. Here we go. Here's the second part of it. I'll just forward it through to you now, Jeff.
So just do a look at the intent. Okay, so the question is, do you know which organic amendments, such as compost, mulches, and biosolids, provide the optimum humus fraction given all the samples your team's looked at? I think optimum humus. One of the things we've got to think about is how decomposed the material has been and how composted. So, and what has been the source materials from which the compost or the biosolids, the organic amendments, have been derived. One of the things that I'd say is, in addition to that, is first of all defining what you want that organic matter to be doing. If you want that organic matter to be enhancing nitrogen supply, then you would like to get a fairly mature compost with a low C to N ratio, so that when that compost gets onto the soil and starts decomposing a little bit itself, that the nutrient release will be high. If you, and particularly then say if it's C to N, if you're adding that material for other reasons, like maybe on a sandy soil trying to open up to hold more water, then you may be more interested in getting a compost or an amendment that is not quite so highly decomposed and can soak up water like a sponge, in essence, and hold it for plants. So my feeling is really when you're thinking of the compost or the organic amendments that you might have available, think through what it is that you really need to get out of that compost, and then what are the characteristics, and I'm using compost there really generally, what are the characteristics you need out of that compost or organic amendment in order to achieve that particular function. Darren's also added on to the end of that, suitable for viticulture. So if you were interested in trying to reduce evaporation by surface application of compost, you would want a different compost than if you were interested in getting it into the soil and having it provide a slow release nitrogen fertilizer. So I would be suggesting that that's the criteria that I'd be looking at, as opposed to me saying which one of these composts is best.
Okay, thanks for your question there, Darren, and your comments around that, Jeff. We don't have any further questions at this stage, but we will stay online for another couple of minutes and just wait to see if we get any final questions.
Okay, so it looks like there aren't any other questions. Thanks for those of you that attended and listened. Happy to take some other questions or comments later on if you want. You can look me up and email me through my CSRL address if you like. Thanks again.
Okay, great. Thanks for that, Jeff. Just quickly wrap up the session. Thank you firstly to Jeff for coming in and presenting today. And a thank you also to everyone who attended and took part in the session. For all the attendees, you will receive a follow-up email which will contain a link to the recording of this session. It'll also contain a link to a survey. Now, the next AWRI webinar is on the 29th of September, despite what it says there on the screen. That's actually incorrect. And it's Anna Carew from University of Tasmania who will present innovative Pinot Noir maceration. If you'd like to register for this webinar and you haven't done so already, please visit the AWRI website. That's all we have for today. Thank you again for attending, and I look forward to seeing you at the next AWRI webinar.